welcome everyone. Welcome to the Royce, but also for a lot of you, welcome to Manchester. And you might be surprised with the weather today because it's absolutely fantastic. And I can assure you that most of the time in Manchester, it's like this, although, and despite what people say. Over the next 10 minutes or so, I'd like to give you a brief overview of the technology platform in bioprinting at Royce. In terms of what is our vision, our mission, the technology that we have available, and also give you some examples of applications of projects that we have been supporting with the technology here in the Royce. So I'm also a senior lecturer at the Department of Organic Aerospace and Civil Engineering, but today I'm just going to put my hats off technology platform lead and talk you through some of the examples that we have here. So very briefly, I know that some of you are already familiar with the Royce. For those of you who are not, the Royce was established with the support of the UK government to capitalize basically on the UK expertise in terms of advanced materials, to try as much as possible to accelerate the translation of products to the markets. The Royce is not just this hub here in Manchester. The Royce is a national institute. We've got several partners across the UK, and we normally say that it's actually a national institute with a regional footprint. So we've got partners in Leeds, we've got partners in Cranfield, Oxford, obviously Manchester, Liverpool, but this is not static. And obviously we welcome always more partners to the Royce. This building was an investment of about 100 million pounds, but I think that more importantly than that is the critical mass that we have in terms of staff. So we've got, the Royce brings together almost 380 people trying to tackle some of the biggest challenges in terms of materials. And obviously these challenges are very different in terms of nature, in terms of scale, but there is a common denominator, advanced materials. And this team, or this group of staff that we have across our different partners work in a wide range of challenges, from transition to zero carbon, to digital and communications, circular economy, so all things that you are probably already familiar with. But probably more important for us today is health and well-being. And what the Royce has been investing, not just in terms of infrastructure, facilities and equipment, but also in people, is to try and support the developments of better, more affordable healthcare therapies, and also personalized therapies. And it was with the aim of supporting that view and that vision of the Royce that we have established the bioprinting technology platform, which I lead. This is a quite unique technology platform, certainly in the UK, but also around the world. And what makes this technology platform quite unique is the broad range of techniques that we have in a single place that allows us to develop, manufacture, and test materials and products for bioengineering. So we've got, as I've said, a wide range of technologies, and some of them you'll be able to have a bit more of contact over the next coming days. We'll also show you some of the other technologies, complementary technologies that we have here. But from 2-photon polymerization to fused deposition modeling or extrusion and laser sintering of polymers and metals, you can have access to all of these technologies, allowing you to work with a wide range of materials, from polymers to ceramics to metals, but also at different scales, from the nanoscale to the micron scale, and at different rates of production. Obviously, the Royce is not, and the bioprinting platform is not completely isolated. So we make part, or we are integrated in a much wider area called the Biomaterials research area that is led by Johnny Blaker. And in this research area, we have different technology platforms. On top of the bioprinting technology platform, we also have the additive manufacturing and printing, looking at bespoke, hard, and soft implants, for personalized medicine. But also we have nanofiber devices with a wide range of fiber spinning technologies, from small scale to large scale production, bioelectronics and smart materials, looking at different things like biosensing, monitoring and stimulation, and a suite that we have on the 4th floor that you also have the opportunity to see during the next couple of days for biomechanical evaluation and testing with a wide range of dynamic mechanical stimulation devices that allow you to apply multimodal stimulation independently of the tissues that you want to regenerate. So I'm not going to spend too much time on this, but obviously I should say that this technology that I'm going to show you over the next couple of slides are the technology that you're going to be using in the next couple of days, but we have a much wider range of technologies available. We have light-based assisted bioprinting. This is an example, the Luminex, which is a digital light processing technology allowing you to use photopolymers for bioapplications. And the advantage of this system is that it's a small-scale system, ideal to test and develop materials with or without the incorporation of cells. And you can incorporate cells in this process because we use visible light. There are obviously different applications for this technology, from microfluidics and vascularization, as you can see here in this example, but also scaffolding, acellular scaffolds for applications in different soft and hard tissues. As I said, there are more light-based technologies like 2-photon polymerization and other systems that we have that allow you not just to develop and test materials, but then to produce at a much higher throughputs, depending on the requisites of your projects. We have also a wide range of extrusion-based systems that allow you to print multi-material, multiple cell line, and multi-scale constructs. These systems allow you to do that because of the wide range of printhead technologies that are available that you can use independently, or you can use them together. So you can select a combination of printers depending on the kind of tissues that you want to create, depending on the type of cells you want to use, and depending on the type of materials that you want to combine. So for just as an example, electromagnetic deposition, so based on inkjet technology for example, cell patterning. We also have pneumatic extrusion for more viscous materials with or without cells. We've got thermoplastics printhead if you want to print, for example, stiff materials. Syringe pump, similar to pneumatic extrusion for high viscosity hydrogels with encapsulated cell. But in this case, you have a much better control over the flow of material, which is important in some of the applications that you have. And then you've got printheads that allow you to cross-link materials using visible lights. So you can use the photochemical heads that we have available. And this allows you to cross-link materials at different wavelengths from 405 to 520 nanometers within the visible range, which for us is critical when we talk about biological applications. And obviously, depending on the polymers that you're using, if they are thermosensitive because of the cells, if they're integrated or not, you can have control over the temperature, not just in the printheads, but also on the building platform. And then you also have a module that allows you to do coaxial. So you can print 2 materials, 2 cells, and you can build, for example, vascular-like constructs. As I've said, this is not the only system. This is a system that you're going to be trained on. We have a much wider range of extrusion-based devices for you to access. Finally, and again, very briefly, I'd just like to talk you through some of the applications, some of the projects that we've been supporting in the platform. Most of these projects, or a great part of this project, is related with skeletal tissue regeneration. This is an example that we have here on the left of a project that we developed in collaboration with Queensland University of Technology to develop stiff bone scaffolds, composite materials for large tibial defects. And I think here on the right, this was a project that we've started developing a long time ago with the University of Utrecht. But I think I've decided to bring this because this is something that we're still supporting here. But also I think it illustrates very well the capabilities that we have and how can you combine all these different printhead technologies to create functional graded constructs. And I think the osteochondral, although challenging, is a very good example of that. And what you can see in this animation is the sequential printing of multiple materials. So a material that resembles the composition and the structure of bone that can be selected automatically by the user. So you have the deposition of the subchondral bone material with or without cells encapsulated. And then you can start layering the different areas of the cartilage. So the deep, the middle, and the superficial. In this example, we use a combination of hydrogels with stiff materials to provide a bit of mechanical resistance. And then obviously, probably the most challenging part that I don't think we've got there just yet is the surgical implantation and the integration of these engineered devices with the surrounding tissues. But this is something that you can do, not just for osteochondral, but if you have functional gradients in the tissues that you're working with, by selecting multiple printing technologies, you can select and combine multiple cells, multiple materials to create these kind of graded structures. Another completely different application that we've also been working with, and this is courtesy of Professor Nigel Hooper's group, is neurovascular engineering. Obviously, progress in the field has been hampered by the limited access that obviously you can imagine that we have in terms of human brain. So we need better models to not just to test drugs, but also to understand the mechanisms associated with the degeneration of, for example, the neurovascular unit. In this specific project, the group was interested in combining multiple cells that exist in the neurovascular unit, like neurons, astrocytes, parasites, combine them with hydrogels, and then print them with bioprinting technology to try and recreate the structural and compositional organization of the neurovascular unit. And then the objective in here was to probe this model and to try and understand, for example, how the accumulation of amyloid beta would promote the disruption of the tight junctions that we have between the endothelial cells and disrupt the blood-brain barrier, leading to a progression or the initiation of dementia. A very recent application that we've been involved quite actively has to do with space exploration. So the European Space Agency has launched a program to develop and install a 3D bioprinting facility in the International Space Station. The ambition is to enable human exploration of deep space. The problem with that is the health of the crew that is very often hampered by some space stresses, like the absence of gravity or radiation. And we don't know exactly the effect of those space stresses on human health. So the goal of this project is first to understand the effects of these space stresses in humans. Because we cannot test in humans, we're trying to develop and bioprint models of humans, just as we do here in the labs, but test them then, fabricate and test them in the International Space Station. The Royce has been involved, or the involvement of the Royce is twofold. One, we're part of a topical team with other partners in Europe to advise on the design and installation of the actual bioprinting device. The other one, we have been selected to integrate this very restricted group of facilities, bioprinting facilities in Europe that are open for researchers that want to develop projects in this area to access and use the technology that we have available in terms of bioprinting. Okay, so I promised I wouldn't be too long. If you want to know more, and as I said, we've got a wide range of bioprinting technologies, but not just bioprinting technologies. We've got a wide range of complementary suites that you can use for your projects, from cell culture, imaging, and testing that you can access. So if you want to find out more, please access the Royce website, or please feel free to email us and sign up for your newsletter if you want to know more about the events that we run like this, but others that potentially are interesting for your projects. And obviously, if you want to follow us on our social media, please do so.